Thanks everyone for being here. Welcome to our Evo Nano workshop. First, I would like to make several announcements. In the first place, the workshop will be recorded, so if anyone would not like to be on camera, you can turn it off. If you have any questions, please use Q&A or chat, and we will answer them after the break. And if anyone wants to tweet on social media, hashtags are are in the chat. And yeah, also we have agenda, so you can follow what is happening. I will start with introductory presentation. Just give me a second to share my screen. Yeah, I think it's fine. So yeah, hello everyone. Again, I will first present you a brief overview of our Evo Nano project, whose full title is Evolvable Platform for Programmable Nanoparticle-Based Cancer Therapies. It is a Horizon 2020 project in the program FET Open. It stands for Future and Emerging Technologies. First thing is, what is actually the problem we are trying to deal with? As the title of the project says, it's cancer, but why it is so important and why we tried to approach it in a way I will explain. First of all, if you look only at Europe, cancer accounts for 20% of deaths each year, but at the same time, clinical approval failure rate for novel cancer drugs is staggering 87%. So we have a huge discrepancy here. We have a very big problem where each day globally 52,000 persons are diagnosed with cancer, and it's a huge loss for society and for persons, of course, but at the same time, things that should deal with it, novel drugs, novel therapies, are not working as expected, and bringing new things to the market is also extremely risky with huge failure rate, and it demands a very long process, and of course it's very costly. So our starting point was to try to imagine that you could test efficacy of nanoparticles on a virtual tumor. And from that, our main vision is to develop digital tumor simulator of the effect of nanoparticles on it. How it can be used? There's a lot of ways how it can be used in the real practice. And I outlined here three most important ones, or three most important in my opinion. First, it could be used to improve lead optimization in drug discovery, because with such a simulator, you can get results that will show you interplay of compound properties of novel drug that you're synthesizing, novel drug delivery system, nanoparticle, or whatever, and their efficacy and behavior can be tested for a specific tumor type. At the same time, it could help in defining optimal patients for a given drug, because if you are able to tweak properties of tumor, then you can see in simulator what properties are important for displaying efficacy of some drug. And both of these things lead to opening possibility for personalized patient treatment, we're using virtual tumors as a platform for running simulation experiments. We can monitor disease progression and metastatic probability for different drugs and treatment regimens, and we can evaluate how they work. The overall pipeline of the project is displayed here. On the left-hand side, there is an artistic impression of the computational model, which is in fact a multi-scale model where we have a tissue scale level, which is depicted here. Below that, we have a cell scale level, and even below that, we have molecular scale level simulations. At the highest level, we can run simulations. We can actually first develop model, run simulations. As a result of those simulations, of course, we go back and forth trying to improve basic functioning of the code. But most importantly, if we are still in the domain of simulations only, we do not know whether they have any sense or not. So that's why in the project, we proceed with synthesis of specific nanoparticles that are suggested by the platform and test their efficacy both in vitro and in vivo. And you will hear details of each of these steps later on. In vitro tests are here to see what are the details of behavior of those nanoparticles in a certain environment, while in vivo tests, of course, are designed to show whether in the real situation that there are some important outputs. Then, since it is impossible to get everything right at the first try, we have built feedback from those experimental results back to the model, where we go back to the drawing table, see what went wrong, try to improve it, and then go again through the entire cycle. Who is doing that? Well, our consortium is composed of 7 institutions, and as you can expect, most of those are in the domain of computing. We have 4 groups in that domain. The first one, me and my group, and we are developing a multi-scale tumor simulator. Sabine from Bristol University is mostly focused on the pipeline for treatment simulations and cell-level simulations. And Adamatsky with his group from the University of West of Finland is devoted to machine learning algorithms and the optimization of drug parameters, because one of the quite important things here is that entire simulation framework is connected with machine learning algorithms that can, on the run, search through optimization parameter space and optimize what is optimize the simulated strategy against the given tumor. Sebastian from Abo Akademi is he is the leader of our group for supercomputer large-scale simulations, because, as you can expect, those simulations are quite computationally expensive. And that's on the simulation part. Synthesis is done by ProChimia Surfaces company, who take our suggestions, generate ideation of how those nanoparticles can look like. They synthesize them and deliver them to in vitro and in vivo evaluation. In vitro evaluation is led by Isabel, who is designing, fabricating, and running experiments on microfluidic devices, and Ibane, on the other hand, is the leader of the group for in vivo nanomedicine testing. So thanks for being attentive to this intro, and now I will give the floor to the next. Let me just stop sharing. Yeah, to the next speaker, who will explain to you in vivo part. That's Ibane. Yes. Yeah.
Okay. So hi everyone. Good morning. So thank you very much, Igor, for your introduction. My role here today will be to show you which are the challenges when we are trying to target and treat cancer stem cells. And before that, I would like to give you an overall idea of what we do and how we do things in our lab. So I'm the leader of a group in the Vall Institute of Research. Vall is a big hospital in Spain with more than 1,000 open clinical trials right now going on. So our objective here is to bring nanomedicines closer to the clinical use. So we do in vitro and in vivo testing using traditional ways of doing this testing in our laboratory. So we do that thanks to our network of collaborators. We are within a Spanish network of nanomedicine, and also we have because we have a developed infrastructure to deal with the in vitro and in vivo cells. And with that, we use specific models for the indications that we have for the nanomedicines. And for instance, in a European project that was Smart for Fabry, last year we got an orphan drug designation for a liposomal product. And last week, precisely, we started a clinical study using iron nanoparticles for hyperthermia treatment. So as I said, we deal with targeted and personalized nanotherapies. In the lab, we are able to synthesize and characterize synthetic nanoparticles based mainly in polymeric micelles, but also using hydrogels. And then we also work with natural vesicles or nanoparticles, which are the extracellular vesicles. And in both cases, we are able to load them with different type of active principles, either drugs and nanobodies, proteins, etc., to drive the therapy into the target cells. What we are bringing here to this consortium and to this project is our experience and testing this kind of nanoparticles in different type of in vitro and in vivo models. And basically, in the case of the Evo Nano, we are using animal models where we can trace the biodistribution of the nanoparticles in vivo and also ex vivo by different type of techniques. But here the objective in Evo Nano was to see if we were able to specifically target the cancer stem cell population within the tumors, and which would be the best design of the nanoparticle that was going to be able to do that. So before reaching to the cancer stem cells, I wanted to give you a brief introduction of why these cells are so important. So as you know, in tumor and in the Evo Nano project, we are dealing with breast cancer, and specifically in breast cancer, not all the cells within the tumor are identical. So we have cancer cells and we have stromal cells, but then within the cancer cells, we also have different type of cancer. Not all are identical. And what happens is that due to this heterogeneity, because we have these selecting pressures with radiotherapy, chemotherapy, and all type of treatments that we are giving to the patients, then we have a situation where, in some cases, we have tumors that are not responding or tumors that are relapsing after the treatment, and this gives rise to the metastases that are actually the final the problem that is actually causing the most of the in the breast cancer patient, at least. So why is that we have this heterogeneity within the tumors? And there are two models explaining this. The first one is the classical one, saying that we have a certain cell that is able to accumulate mutations during the progression of the disease, and that this runs different type of tumors that then you need to treat with different type of treatments. Opposite to this, what is more accepted now is that what we have at the very beginning is a cancer cell that is kind of a stem cell, and this is why we have this name of cancer stem cell, and that is able to maintain these stemness properties and give rise to cells that are also differentiated. So in the first case, if we are treating the tumor, we have different therapeutic targets. But in this second case, where we have the cancer stem cell theory or hypothesis, by targeting the cancer stem cells, we would be able to get rid of the whole tumor. And this is what we are trying. The cancer stem cells, as I said, are very scarce, low number of cells within the tumor. Depending on the tumor time, they can go for 0.1 to 10% of the cells within the tumor, and they have this ability to self-renewal or differentiate. And because of that, they are able to initiate tumors when they are inoculated in very small numbers within the animal models that we are using. And not just that, but they are also more able to migrate and metastasize compared to the differentiated non-cancer stem cells. One of the main features of these cells are they are growing very slowly, so the conventional chemotherapeutic drugs are not being active in this kind of cells, and also they have overexpression of certain transporters and other mechanisms that makes them very resistant to the conventional treatments. And so these two things, the low number of the cancer stem cells within the tumor and their ability to resist the conventional treatments, are the two challenges that we need to face when we are dealing with cancer stem cells. So this is just a scheme of which can be the mechanisms by which the cells can be resistant to chemotherapy. They can either, as you can see here, overexpress certain transporter that take out the drugs, the conventional drugs. They can also overexpress different type of detoxifying enzymes that help the cells get rid of the drug. They can avoid expression of certain molecules, such in the case of the breast cancer, the expression of HER2 receptor is used for targeting therapies to the cells, but cancer stem cells may have very low levels of this receptor in the membrane. They can also be more resistant to the apoptosis or to certain type of cell death. So knowing this, the idea is that we can use different strategies to overcome this resistance and to actually target the cancer stem cells. One would be to use specific drugs that are able to target the pathways that are very active within the cancer stem cells, or drugs that are able to differentiate cancer stem cells to non-cancer stem cells, and then conventional therapy will be useful and feasible in those cells. The second mechanism is to try to overpass the drug resistance by using nanoparticles. Why? Because in this case, we are delivering the drug within a system that is going to introduce the drug and put it far away from the membrane where the drug transporters are. By this, we can increase the efficacy of these drugs. And the third mechanism will be to use specific targeting moieties to specifically arrive to the cells that are the cancer stem cells and increase the loading of the amount of the drug that are actually reaching those cells. So in our case, in our lab, we are trying to develop these three strategies by different mechanisms. And for that, we are using an in vitro model where we can trace actually the expression of the quantity of the cancer stem cells that we have within the cultures. How we do that? We do that by using a promoter that is specific of the cancer stem cells linked to a fluorophore, which is the T2. In this way, we can see within a culture which are the cancer stem cells and which are not the cancer, which are the regular cancer cells. With not just that, we can also sort cells and have cultures with just cancer stem cells or with the differentiated cells, and we can also validate that this is not just something that we can see by image, but we can also extract the RNA or do the functional assays in vitro to verify that we are actually having those stem cells within the culture. Using this model, then we can test nanoparticles and see if they are effective or not. And this is what we did at the very beginning, just to select the drugs that we hope we searched first in the literature that they were going to be active in cancer stem cells. So what you have in the table is a list of different small drugs, and we determined the IC50 value, the concentration of the drug that was able to kill half of the cells for the TD tomato positive for the cancer stem cells or the TD tomato negative non-cancer stem cells. And there we could identify which were the drugs that had a lower value of the IC50 in the cancer stem cell population. And for instance, the niclosamide and the 8-quinolinol that are further developed in the Evo Nano project were identified as drugs that actually had lower IC50 values for the cancer stem cell TD tomato positive population. We also know that we need to target not just the cancer stem cells population, but also the regular cancer cells. And for that, we developed different assays where we could use the combination of a conventional chemotherapy, which is the paclitaxel, PTX in this graph, and the two drugs that we selected, the 8-quinolinol and the niclosamide. And what you can see in the graph is that when we are using the combination of the both drugs, the number of cancer stem cells within the culture is drastically reduced. So eventually, in the clinical setting, we are thinking about the situation where we are combining those nanoparticles delivered in the niclosamide or the 8-quinolinol along with the conventional paclitaxel treatment. An example of a nanoparticle that we used to test the efficacy in the cancer stem cell model are these polymeric micelles containing citrol. Citrol is a drug that is active against cancer stem cells, but it's very unstable. So in this case, the nanoparticle or the use of the polymer was there to stabilize the drug. But then we realized that when we were treating the cancer stem cells, the TD tomato positive cells, what you can see in here, the percentage of the TD tomato positive cells in the culture was being reduced, so meaning that the polymeric micelle, the CML, the second bar in your graph, was actually being more active than the free drug. But we can also use the same system or a similar system to target it. And in this case, we developed a nanoparticle where we were including paclitaxel, this conventional chemotherapy against breast cancer, but this nanoparticle was targeted to the CD44. CD44 is an antigen that is overexpressed in the membrane of the cancer stem cells. And by using this, we realized that when we were using the system that was including the CD44, this is the light green line in here, by the dose of 0.2 micromolar of paclitaxel, we could get rid of almost all the cells in the cultures. But when we were using the same system without the CD44, then we were not killing that many cells. And why is that? Because this nanoparticle is able to reach better to the cancer stem cells. And not just that, but also because when we are using the free drug, the quantity of the cancer stem cells that we detect in the culture is increased. This is what you have in the second graph in here. When we are treating the cells with just the free drug, the percentage of the cancer stem cells is increasing compared to the control. But when we use the nanoparticle that is specifically targeted to the cancer stem cells, this increase in the percentage of the cancer stem cells is being reduced. So we are able to counteract the effect of having a drug that is inducing the increase of the cancer stem cell population. And lastly, the what we are doing is just trying to combine these two strategies, selecting a specific drug, which in this case, in this example, is the niclosamide, with a nanoparticle that is going to be functionalized with a targeted moiety. And in this case, the targeted moiety is also against the CD44, but to a specific isoform of the CD44, which is the CD44b6, by using a FAB, a small antibody. And using this system in here, in vitro, we saw that the efficacy of the targeted system, again in here, in the first graph, is higher. And when we moved in vivo, we saw that even though in the primary tumor we had some effect, this was not that much. The volume of the primary tumor is smaller when we are using the nanoparticle. Where we could see a huge effect was in the amount of circulating cancer cells. So somehow these targeted nanoparticles are very effective to reduce the number of the cells that are going to give metastasis later on. But still, there are many questions open. So it's very difficult for us, by using the systems that we are showing you here in vitro and in vivo models, to determine which is the dose and which are the best combinations between the drugs. It's very difficult to us also to understand the dynamism between the change from the non-cancer cells to non-cancer stem cells to cancer stem cells, this interplay between these two stages, and then the interaction of those cells with the surrounding stroma, the endothelial cells, the fibroblasts, etc. Because there are many players, and when we do in vitro testings with our regular cell line, it's difficult to incorporate all this information, and also it's very difficult to us to predict how a nanoparticle is going to behave once we are administering it in vivo. So we often go to the in vivo testing without having a real clue of what's going to happen with the nanoparticle. So in our case, specifically, we are trying to use, as I told you, the combination of paclitaxel with echinolidone and niclosamide in nanoparticles, because we think that this is going to be a way of getting rid not just of the cancer stem cells, but also the regular cancer cells. We are also know that in this dynamism from being cancer stem cells to non-cancer stem cells, in this differentiation process, the extracellular vesicles are very important. So we are also trying to dissect the role in this process, and we are also trying to use to identify and to develop a specific nanobodies against the cancer stem cells to be more to have better tools to target those cancer stem cells. And we are doing that thanks to the Evo Nano project, but also thanks to additional European projects such the SAFE and MedTech and the MIMIC-KEY projects. And this is it. This is the team and the funding agencies. If you have any questions, I will be happy to take them after the break, or I can also try to answer you through the chat. Thank you very much.
Great, thank you. I think I'm up next. Great. So hello, everyone. I'm Dr. Sabine Hauert. I'm an associate professor of swarm engineering at the University of Bristol. And the focus of my talk is going to be on the automatic growth of tumors and the design of nanomedicine to treat those virtual tumors. So nanoparticles, as we've heard in the previous presentation, have the potential to be useful vehicles for the delivery of drugs directly to certain types of tumors. And these particles work in huge numbers, in the 10 to the power 13. And so it's critical that we understand what the collective of these nanoparticles are doing so that we can optimize their impact on tumor tissues. And what fascinates me is that there's many different ways in which you can design a nanoparticle. You could change its size, that will impact how well it's able to move through tumor tissue. You could change its shape. You could change its charge. You could change its material. Some of those materials might react to their environment or be energy receptive to light or to magnetic fields. You could decorate these particles in a way that allows them to target receptors that might be overexpressed on certain tumor cells. And you can load them with different payloads, as we heard in the previous, which will have different impacts on the tumor environment. And what's fascinating is that the way you change all these aspects, the way you turn the knob on the design of these nanoparticles will change how all those particles, the 10 to the power 13, are working together to treat a tumor. And indeed, those design parameters are going to change the ability of the nanoparticle to be transported to its site of action and to overcome transport barriers within the body. And that includes their ability to move through the bloodstream, and then from the bloodstream to extravasate, so to leave the bloodstream to enter tumor tissue, to move through that tumor tissue, and then to be able to be uptaken by the cells and act on the area that we want these drugs or payloads to act. So what we've been asking within our team is, how should we design the nanoparticles to get the best tissue penetration? And this is just a thought experiment from a while back, from 2013, where we were looking at two knobs, two parameters on the design of the particles. For example, its size, which changes how fast they move, and their binding affinity to receptors on certain cancer cells. And so if you have a vessel and the particles leak out of that vessel, how should you design those two parameters so that they can accumulate at lethal levels to kill those cancer cells up to a certain depth? And indeed, mathematical modeling allows you to wrap your head around this question. So we can inject virtual nanoparticles with a certain drug, with a certain lethality. You can look at what percentage of that drug ends up in the tumor, and then you can zoom into the tumor here and look at what's the hardest situation to tackle. And for example, it might be the ability from one vessel to go as deep as you can from any vessel point in that tissue. So for example, we might want to go 20 cells deep, to give you an indication of this, which corresponds to around 200 microns or so. And so at the time, what we thought was interesting is that just changing those two knobs, so your ability to move fast or slow and your stickiness to the cancer cells, entirely changes where the nanoparticles distribute over those 20 cells deep within the tumor tissue. And indeed, some of these particles sounded great. A, for example, was a particle that was very good at sticking to cancer cells and getting eaten up by them. But if you look at what what all those particles do, what happens is they leak out of a vessel, all stick to the first cells they encounter, and as a result, don't go deep into the tumor tissue. So while the design of the individual particles made a lot of sense, the design of the collective did not. And just those two changes, those two knobs, give you this whole range of different kinds of distributions within these tiny chunks of virtual tumors. So since then, as part of the Evo Nano project, we've really refined our simulations. So we're now able to capture space more efficiently, and we can model nanoparticles with their receptors. We can model the way they move through these tumor tissues using what we call stochastic models. And then we can visualize some of these things in Tumours-on-a-chip, and you'll hear much more about this from Isabel later in the presentation. And so we have ways to create better models of these little tumor chunks and to validate that with in vitro simulations. So that led us to this design of a framework amongst the whole cohort here. And the idea was, well, if you could specify the properties of a tumor and what types of payloads you think would be useful against that tumor, then could you virtually grow a tumor with those characteristics? And then what you would want to do after that growth is basically carve out lots of representative tumor chunks, those little tumor chunks, And those little tumor chunks would then be used as a playground for machine learning to automatically design nanoparticles that are good at treating these virtual tumor chunks and hence the overall tumor. And further down the line, as Igor mentioned, it would be interesting if those specific tumor properties came directly from patients, and maybe you could personalize them and grow digital twins of patients, individuals' tumors, so that you could optimize the treatment that would work for them. So if I give a little bit of a zoom in, this is what growing virtual tumors looks like. This is done by Igor Balaz's team, working with PhysiCell, which is an agent-based model, and you can specify the rules for the tumor growth. Let these tumors grow, and so this is what it might look like at 7 days. And then you can see there's different colors here. This is a simple tumor where you have vessel points, you have cancer cells, and you have necrotic cells in hypoxic regions. And we can carve out these colors. Here we're interested in seeing where the vessel points are and the cancer points are. And we do statistics to then generate lots and lots of little tumor chunks, which is what you see here. So these little tumor chunks then serve as our playground. And this is what machine learning looks like in this context. What we're trying to do is optimize the nanoparticle design, and the knobs that we're turning have to do with the speed or the diffusion, the dissociation constant, so how well it is able to interact with the cell, the dosage you inject, and how many drug payload you have per particle, so how much it's carrying in each one of those particles. And here what we call generations, you can think of this as our ability to go from not having learned what's the best particle to having learned what our best particle is. And here what you can see is sort of the trajectory. So at the start, we start with random particles in simulation and test them in our playground. And you can see initially here, to get a better performance, we want to increase the diffusion so they can go deeper. We might decrease the binding, the stickiness, so that they don't stick to those first cells. And you can see as we go along, we might actually decrease the number of drug molecules or decrease the nanoparticle concentration. And the reason the machine learning does that is because we're trying to optimize two things: the number of cancer cells killed and the overall, reduce the overall injected dose to avoid side effects due to damage to healthy tissue in the surroundings. So the learning process is doing that. It's tweaking those parameters and it's reducing the overall injected dose. Now we can use this platform as well to think about more sophisticated scenarios. So this was the case where you just had cancer cells, and you can see that if you just look at NPI, that means internalized particles. Our internalized particles are just above the lethal dose. So this has been nicely optimized so that you can kill all those cancer cells here. These are all different cancer cells, and you kill them with just enough drug. So that's a case where we've optimized the dosage and the type of particle. Or you can put more sophisticated treatment tumor profiles. So here Igor's team has grown tumors which include cancer stem cells here amongst the cancer cells. And what we're optimizing here is how do we combine two particles, a bit like you heard from Ibane's presentation, so that we have the right number of particles treating the cancer stem cells as well as treating the cancer cells, and what strategies would give us a good outcome. So we found that particles that diffuse well and have the right dissociation constant is one way to go, but we also had a different strategy, which we hadn't thought of, that had to do with lower diffusion. And so sometimes it's interesting to see these trade-offs that then bioengineers like ProChimia would need to go and design the right particle with those characteristics. So just to wrap up, we're currently designing an online tool. So this platform is open source. You're welcome to download the code already. It's part of the paper. So feel free to go to the paper, download the code and have a go at it yourself if you'd like to optimize your own nanoparticle treatments. But coming soon, we'll have an online tool that should allow you to do this using a web browser. If you have a little bit of patience and wait for an email to come in your inbox with the result of those simulations. So this is a very cross-disciplinary effort with our team at University of Bristol, but the whole Evo Nano platform builds on work from across the board. So all the people that you'll see here have contributed to making this cross-disciplinary and multi-scale platform to optimize nanoparticle treatments. So thank you very much. And our next presenter is going to be Antti from University of West of England. And he is going to tell you about how more sophisticated machine learning frameworks work to optimize nanoparticles.
Thank you, Sabine. Let me just start sharing. Okay, thank you for coming. On my turn, I would like to welcome you. My name is Antti, and I'm a lecturer on computer science with the University of the West of England. My part as a member of the Evo Nano consortium was being involved with the designing and testing of possible optimization methodologies for the platform. And as you heard previously, cancer tumors are very complex systems. They are comprising of multiple different types of cells that they can establish their own microenvironment. Also, there are complicated communication pathways between different cancer cells that can lead them to react and adapt to different situations. So, for instance, a simple treatment may cause drug resistance after a while. So a possible treatment needs to be carefully designed with appropriate complexity to match the complexity of the problem. And it would be beneficial if the treatment would be able to respond to this adaptability of the problem of the tumor to drugs. So also the designing of treatments is not an easy thing. As Sabine showed previously, there are several parameters that need to be checked. So there is a large set space of parameters that needs to be carefully thought of. So facing such a problem with high complexity, the way forward is producing computer simulations of the problem and applying optimization methodologies like evolutionary algorithms. So evolutionary algorithms are inspired by biology and in specific evolutionary theory or genetics. And what they do is employing an initial population of solutions, which after mutation and crossover between them, there are fitter individuals produced for the given problem. So to study this automated optimization methodology of the problem of designing nanoparticle-based treatments, we used an established simulator of a tumor microenvironment, Physicell, which is open access. You can find the whole code on the cited web page on the slide. So as it is an open source tool, it was easy for us to adapt it in our needs and use it as a test bed for several optimization methods. So the simulator is agent-based. So by setting different parameters on the simulated treatment, in the video represented by the blue and red dots or agents, we have different outputs in terms of the remaining green dots, which represent the cancer cells. So these outputs, the remaining cancer cells, are considered as the fitness of each treatment or the set of parameters that we give as an input to the simulator. So by using these numbers as fitness functions, we applied several different optimization methods, from conventional and well-established ones to more unconventional like the open-ended novelty search, or really powerful cutting-edge methods like surrogate-assisted models. So surrogate-assisted models use AI models. They train them to react in a similar way to the testbed or the simulator, but they use these trained AI models in order to guide the optimization process because they are less computationally demanding than the actual simulator. So there's not enough time to go into details, but you can refer to the cited works on the slide. You can visit the project's web page where there are links to all our works, which are all open access. And after dealing with different types of optimization, what we thought a good idea would be is to try not only to find the best design for one type of nanoparticles, but it would be interesting to find how different types of nanoparticles used within the same treatment will affect the fitness of the treatment as a whole. So as a result, we made appropriate changes in physical's code to simulate multiple types of nanoparticles within a single treatment. And we employed specialized evolutionary algorithms where the length of the best solution is not known beforehand. And by length here, we define the number of the different types of nanoparticles. So we implemented evolutionary algorithms with metameric representation, and we used appropriate mutation and crossover operators in these algorithms. Also, we used another parameter, which was the application time or the release time of each different nanoparticle, but within the same treatment, so within the same simulation. And in a way, we wanted to see if there is some kind of multi-stage dynamics of the tumor within the simulator, and if these dynamics could be tackled by a multi-stage release of different types of nanoparticles. So by observing the results, I will not present them now because of the tight schedule, but you can refer to the works cited. We concluded that while the simulator did not include some predefined dynamics on the tumor development phase or mechanisms on drug resistance, there was actually an improvement on the multi-phase release of different types of nanoparticles when the timing of this multi-stage release was indicated by the automatic optimization method. On the contrary, when the same nanoparticles with the same parameters were applied all together at an early time slot, the results were not as good. So thank you for your attention and your time. Please refer to our webpage where all the information are available. And I would like again to acknowledge our funding agency. And I would like to stop sharing, and I think Igor is next with slides on growing in the virtual tumors.
Yeah, hello everyone. I will be talking next. Let me just share the screen. Yeah, so I'm Oksana Havryliuk, and I will be talking about AI-based molecular modeling as the part of the Evo Nano project. And I first wanted to introduce our team, because without them, all of these results wouldn't be here. My team is Poonam, Rizal, and Rafo, and me, Oksana, presenting. Also, we have Sepinot and Sebastian as our supervisors, and some special regards to Victor and Otto, as without them, they also contributed to this project. And I wanted to firstly do a small introduction on the data that we are using to define a clear goal of our project, and then we'll move on to the approach itself. So for evaluating nanoparticle design, our team is using efficiency matrix SASA which is solvent accessible surface area, and it's important for the drug delivery. After a nanoparticle is created, it takes some time for it to reach a stable state, and the SAS amount drops by that time. We are using the data from the molecular dynamics simulations. The goal of the molecular dynamics is to monitor the change of the efficiency metric in a computer-based environment. It is an affordable solution, but it still takes weeks to go through it. And our team is introducing an efficient way of applying machine learning to predict the stable state of the nanoparticle design and its efficiency metric. In that way, the approach would massively reduce that simulation time and complexity to help the nanoparticle scientists deliver new cancer cures faster. After implementing a model, we will use explainable AI to get valuable insights without any doubts on the decisions that the model is taking. So choosing data processing technique is descriptors. Descriptors account a nanoparticle into a simple structure. They make sure that each design of the nanoparticle has the same representation, regardless of its rotation or permutation. With the help of descriptors, it is possible to feed such a big and complicated nanoparticle structure into the machine learning model. Our team is using a descriptor called many-body tensor representation, or just shortly MBTR. MBTR descriptor can extract the global properties of the nanoparticle using different element pairs and their interatomic distances. For implementing it in Python, we chose the describe library. We propose an approach based on two machine learning models. For the first one, we have developed a time series model to predict the movements of the nanoparticle based on its previous positions for as many time steps in the future as required. The model is based on the window approach, where we input 40 MBTRs to forecast the 31st one. Then we use attention-based transformer model for MBTR forecasting. As the data for the training is also derived from molecular dynamics simulation, we had very limited data to get proper accuracy with such a large model as a transformer. But as we had MBTR vectors representing different physical systems, through probabilistic models, we could synthesize a lot more data and improve the model's accuracy. So here you can see the model's performance, where the blue bars represent ground truth values and the red ones the predicted values. And you can see by the graph that the model is pretty accurate. And then, based on the structure of the nanoparticle, it is possible to predict the efficiency metric, in our case, SASA. And the second machine learning model maps that predicted MBTR from the previous model to the corresponding SASA value. And for that, we used an LSTM-based neural network. Here you can see the model's performance on this graph, where the blue line represents real values and the orange one, the predicted values. And you can see also that the accuracy is pretty high. This slide shows the chosen approach workflow. So first, we have the batch of nanoparticle designs that is given as input data. Then they are transformed into MBTRs as the data processing technique. Then data is input into MBTR to MBTR forecasting model that can predict the MBTR vector at any future time step, which then can be calculated to the corresponding SASA value by using the second model. And the overall system works with a very acceptable amount of error. And the problem with the deep learning model is that only the conclusion is seen, not the output between levels. And as a result, we have no idea how the nodes are analyzing the data, and that is the AI black box problem. And here, the explainable AI is an AI tool that produces results for humans to understand and explain a certain predictive model. We use the shop values, and with their help, we could gain some insights of the features that are the most important for our model's output. And here you can see the bar plots, the sample bar plot with some feature importances. And that's it for my part of the presentation. And then I will welcome the next presenter, the Dr. Isabel Rodriguez from the IMDEA Nanoscience to talk about Tumour-on-a-chip, the screen nanoparticles.
Thank you, Oksana, for your introduction. My name is Isabel Rodriguez, and I come from IMDEA Nanoscience in Madrid. And today, I'm gonna put this, I will talk to you about our work on designing and fabricating Tumour-on-a-chip devices. I'll pass through, sorry. So as we have heard today, nanomedicine or nanoparticles have a number of advantages. These include the improved viability due to the long circulation times in the body. They also they are able to provide a localized delivery to the tumor location. They can be functionalized to do target delivery, and as well can be designed to do control release, as we have here this morning. All this will lead to increase of efficiency of the nanomedicine and reduce the side effects that traditionally are obtained due to systemic chemotherapy, the current treatments today. But to reach good therapeutic outcomes, the nanomedicine needs to reach the tumor and cross these transport barriers. Typically, nanomedicines are applied through the bloodstream. They move through circulation, and they have to extravasate into the tumor environment and later penetrate and internalize finally in the tumor cells to be effective. However, nanomedicine hasn't reached the expectations yet, and this is possibly because of poor delivery into the tumor cells. According to some studies, particularly this by Wilhelm, it's been calculated and that the injected dose that reaches the tumor is only 0.7%. So that doesn't allow to reach high doses into the tumor. And one of the problems for this is because the current models available today do not allow to test, current experimental models do not allow to test the transport properties of nanoparticles. These models, including the traditional 2D cell cultures and animal models. So this leads to a poor clinical translation today of nanomedicines. So what we have been working on is on making a Tumour-on-chip model to recapitulate the tumor microenvironment into a microengineered device. The tumor environment is shown in this image here, is a densified extracellular matrix where microvessels go across, which are leaky. They're abnormal and have some bigger pores than normal vessels. But additionally, because of the growth of in the tumor, there is an extra pressure called tumor solid stress. And because of the poor drainage, there is this interstitial fluid pressure. All together makes the tumor environment to reach higher pressure than the blood pressure. And as such, the transport of nanomedicines from the bloodstream to the tumor is going to be only limited to diffusion, no convention, because of this high pressure. So in our lab, what we do is to build these devices where we try to recapitulate some of these physical parameters, including this bloodstream where nanoparticles are flown through at the blood at similar physiological parameters and at similar shear rate. And we also mimic into the devices the solid tumor pressure, that, because it's an important parameter on the nanoparticle delivery. And we also incorporate in these devices tumor and the endothelial cells to make the endothelial barriers. And one important point is to have build the devices so they can be monitored by microscopy, all these processes. So this is a drawing of one of the devices we made. So typically it's a microfabricated substrate which has several channels and typically will include a microvessel where the nanoparticles are flown through. It also includes a tumor compartment where the extracellular matrix is positioned, collagen or matrigel, and in some of the times tumor cells or spheroids can be cultivated here. There is another chamber, which is the pressure chamber, where we use it to mimic the solid tumor pressure. And the important part here is we have the microvessel is has a communication to this tumor compartment because we have some spaces left here through a micropillar array or blocks. So in the microvessel we cultivate the endothelial cells to make this endothelial barrier that I will later talk more about. So as I mentioned, the one point that we want to recapitulate on the device is the solid tumor stress. So this will be applied through pressure to this membrane, and a video that shows how this how the effect that this produces on the extracellular matrix is shown in the video, compressing the extracellular matrix. An important feature on the devices are the endothelial barrier, which across the nanomedicines or nano materials diffuse across. So we pay attention to have a complete endothelial barrier from the vascular channel to the extracellular tumor compartment. So here you see images of this barrier. These are endothelial cells stained with fluorescent dyes. So you see from the top to the bottom of this microchannel the cells, and in the middle is only the vertical image of this endothelial barrier. And this is an expanded image of the barrier, and in red you can see the nanoparticles associated to this endothelial cell. And we can see nanoparticles on both sides of the barrier. So the devices, we use them primarily to study diffusive permeability. So here are screenshots of time-lapse images of nanoparticles permeating across this endothelial barrier I showed you before. So this will be the endothelial cells, and we have two channels here. One which has a closed barrier, porous barrier of cells here, and here will be a complete enclosed endothelial barrier, and we see the nanoparticles that are flowing through this, the red color that you see, how they diffuse over time. And we see the porous barrier allows diffusion much faster than when we have a closed Hubert barrier on the channel. We calculate is possibly in these devices the diffusive permeability, and it comes to this permeability when we only have collagen matrix is about 7.71 micrometer per second, and it reduces quite a bit when we have endothelial barrier porous or totally closed. And this is a graph representing this reduction. We can also, in the devices where we mimic the solid tumor stress, measure this effect on the transport of nanoparticles. So in this case, we have the device here filled up with matrigel, which is peptidic matrix and has some pores where nanoparticles can permeate through. And we activate, so we pass through the central channel nanoparticles, and we monitor over time the diffusion on the gel. You can see the images here over time, how the nanoparticles diffuse on the gel. And we do this without applying the tumor pressure, and also by applying pressure on the gel, which causes the densification and possibly closing of the pores on the gel. And we can also calculate this the diffusive permeability in both scenarios. So, for example, we can see that when there is no pressure applied, the transport of the nanoparticles tested, in this case at 2.5 nanometer nanoparticles, it moves like 30 nanometers per second. But when we apply pressure, the mobility will be only 10 nanometers per second. So this will translate to reduction on the time that takes to cross the nanoparticles for the matrix within the tumor. For example, in an area of 100 micrometers, that would mean a difference from 1 to 3 hours. Okay, so this is a summary of the short presentation of our Tumour-on-a-chip devices, and I show you that we're able to create a more precise environment of the tumor, and we can predict some dynamic transfer parameters on the device, and it also allows easy and direct observation through microscopy. So where we can measure extravasation and diffusion permeability, and if we could grow on the device tumor cells, we could also measure the efficacy and toxicity of these drugs. So this is the team at IMDEA. It's formed by Alberto, who's a physicist, myself, pharmacist, and Sergio, which is a biologist, and used to be Jan Cacho, which used now in Tokyo University. So now I would like to thank you for your attention and pass to the next speaker, which will be Dr. Dariusz Witt from ProChimia.
Thank you, Isabel. So now I will share the screen. All right, my name is Dariusz Witt. I am from ProChimia Surfaces company, and I give you a speech about the design of the nanoparticles. General ProChimia Surfaces company was established in 2001 and is localized in the north part of the Poland, as you can see over here at the Baltic coast. So the team is consisted of 9 persons and 7 with PhD and 2 professors. And we have customers around the world, and some of them are from the universities and also from the companies as well. And we provide chemicals and nanoparticles to all of them. So in fact, we used to produce chemicals for the functionalization of the variety of the surfaces and formation of the self-assembled monolayers. And also we provide already functionalized nanoparticles, where gold nanoparticles are one of them. And also compounds which are required for the functionalization of the surfaces of some inorganic materials as well. And also the advanced organic synthesis is one of the part of our work. Our company is involved in several European Union projects. Already some of them were over, like Graphinery, manufacturing, Evo Nano currently, and also hygiene project where we are involved. First of all, why gold nanoparticles are selected as the carriers for the active pharmaceutical ingredients. First of all, we can prepare them with variety of the shape and size. We know very well how to perform the functionalization of these nanoparticles by means of the several tiles, which can be terminated with other functional groups, which can improve, for instance, solubility of these nanoparticles under aqueous conditions. And the terminal functional groups can be useful for attachment of the active pharmaceutical ingredients. And first of all, we can also control the size and monodispersity of these nanoparticles. It means that these nanoparticles will be almost equal to one another. So in this way, we do not have to just discuss how the final effect reflects distribution of different size of the nanoparticles included in the examined solution. Also, these nanoparticles, they allow attachment of the several different compounds at the terminal part, so they allow control the final properties well, in the reasonable way. Also, the metallic core is when it's constructed from the gold is with low toxicity, so there is no side effect with the potential presence of the gold in the cell. Also, gold nanoparticles are can be produced based on the good manufacture practice, so they are quite reliable. So there are in fact known 2 major methods for the reliable synthesis of the gold nanoparticles. Each of them requires the precursor of the gold in the form of the salt, and the appropriate stabilizer is required just to control monodispersity of these nanoparticles. And definitely a reducing agent is required to convert appropriate salt into the metallic gold. So as you can see, the first method, Turkiewicz synthesis, is based on the reduction of the gold salt by means of the sodium citrate. And in that case, the citrate, in fact, reduces appropriate gold salt into the nanoparticles and also provides the source of the stabilizer, so which is based on the electrostatic interaction of the gold nanoparticles with the citrate. The second method is performed in the 2-phase system, and the gold nanoparticles are stabilized later by the presence of the appropriate tile, and the reducing agent is sodium borohydrate. Anyway, both these methods are currently the most common methods for the reliable synthesis of the gold nanoparticles. And the stabilizing agent is really important because it allows you to control the monodispersity of the nanoparticles, and also how efficient this stabilizing agent is will be reflected in the final size of the gold nanoparticles. Well, the most common is used sodium citrate. Also, phosphines or ammonium salts can be used for that type of destabilization. And another example of the stabilizing agent are polymer-based compounds. And as you can see, they can stabilize nanoparticles, but they are frequently not very useful because after stabilization of the gold nanoparticles, they do not allow a convenient further functionalization of these nanoparticles. And the most important, in my opinion, are thiol ligands, which are responsible for the stabilization of the gold nanoparticles. What you can see, these nanoparticles, where they are stabilized with the thiols terminated with other functional groups like carboxyl group or amino group or ethylene glycol polymer, they provide appropriate charge at the surface of the gold nanoparticles, so we can control also their final solubility under aqueous conditions and final properties, like for instance internalization as well. Well, so in the case of the gold nanoparticles, there are a couple of parameters which can be very useful. For instance, if you are going to control the size of the gold nanoparticles, you can control the rate of the reaction. So if the reaction is faster, then the appropriate gold nanoparticles will be smaller. So it's reasonable, because if you perform a reduction of the appropriate gold salt in this solution, and this reduction will be performed very quickly, then it means that these nanoparticles, they will have not enough time to grow into the large size. So that's why small size of the gold nanoparticles will be obtained. Also, the amounts of the stabilizing agent is important, because if the amounts of that agent will be high, in that case, in fact, if the small nanoparticles will be produced, they will be surrounded by the stabilizing agent, and it will just stop the further growth of these nanoparticles. That's why smaller nanoparticles will be obtained. Also important is to control that each nanoparticles will be the same. So it means that the monodispersity of these nanoparticles will be very high. So in the case of the temperature, as you can see, at higher temperature, when you perform this reduction of the appropriate gold salt, in that case, more monodispersed particles will be obtained. Also, important is how strong a stabilizing agent is. If the stabilizing agent is pretty weak, in that case, it will be simply, if too small nanoparticles will be surrounded by this stabilizing agent, in certain time they can just be released, and again they can grow up, and they, in this case, monodispersed particles will be obtained. The most difficult part is just to control the shape of the gold nanoparticles. So in that case, we have to use the different stabilization of the crystal faces of the nanoparticles. So in that case, we can obtain, for instance, the appropriate gold nanorods or bipyramidal shape as well. So over here, there is example which I have found in the literature, which clearly expresses the presence of the thiol in the process of the reduction of the gold nanoparticles. The first picture, which you can see on the left side, is the gold, the mixture of the gold nanoparticles, which was obtained during the reduction of the appropriate gold salt and without any presence of the thiol. It means stabilizer. So in that case, as you can see, some of the gold nanoparticles are really pretty small, but some of them are really huge. So from the practical point of view, such mixture of the gold nanoparticles is useless because they are not monodispersed. So it's difficult to describe their behavior, their diffusion. They well, some of them will be moved much more faster, some will move much more slower. Also, the internalization of these nanoparticles will be really different, and from that point of view, the final results will be just really complicated, and interpretation will be difficult. However, when you take a look on the picture of the right side over here, as you can see, in the presence of the dodecane tile, the nanoparticles are obtained almost in the same size. So definitely, as you can see, these nanoparticles are with 6.2 nanometers, and all of them almost are with the same size. So the conclusion is, if we are going to prepare appropriate gold nanoparticles for the biological studies, definitely we have to base the preparation of these nanoparticles in the monodispersed way to obtain all nanoparticles almost with the same size. Over here, there is example also from the literature. So as you can see, the spherical shape, these nanoparticles are can be prepared in the easiest way. In nanorods, as you can see, they have the shape is different, and they can be also obtained, as you can see, and also nano bipyramids can be obtained. Definitely, as you can see, the transportation of these nanoparticles will be difficult to describe. However, spherical shape from the practical point of view is the most convenient at the starting point. Definitely, in the case of the gold nanoparticles, there is no typical chromophore which you can observe for organic compounds when you measure the UVV spectrum. In such case, for gold nanoparticles, where there is no organic chromophore, we shouldn't observe any UVV spectrum. However, we should keep in mind that in the case of the gold nanoparticles, there are a lot of the electrons localized in the gold metallic core. So these electrons can resonate with the appropriate light. So that's why the absorption around 200 or a little less is observed. And what you can see over here at this picture, different spectra are recorded according to different size of these nanoparticles. The only problem is that this technique is maybe just only partially helpful, because it's easy to find difference between nanoparticles with 5 nanometers or 50 nanometers. But difference in the UVV spectrum between nanoparticles with like 5 nanometers or 8 nanometers cannot be observed at all. So the only technique which is precisely enough for determination of the gold nanoparticle site is TM. And these pictures, which you can see over here, they just showed you how the size of the and the monodispersity of these materials can be determined. Anyway, in general way, for the preparation of the gold nanoparticles, we will start with the appropriate gold salt, and then it, in the presence of the stabilizing agent and reducing agent, it's converted into the gold nanoparticles, which are stabilized with the appropriate ammonium salts. And then these ammonium salts, they can be simply replaced by appropriate tiles with the functional groups at the end. And then appropriate functionalized gold nanoparticles can be obtained, and further ligand exchange provides you with the appropriate gold nanoparticles, which we might be interested in. Also important is what kind of the tile you are going to use to stabilize appropriate gold nanoparticles. If we consider the biological studies, then we have to keep in mind that final construct, which will contain appropriate tile, which will stabilize gold nanoparticles and prevent the aggregation of these nanoparticles, should allow also the solubility or improve solubility in the water. So that's why, for instance, frequently carboxylic group or ammonium group can be selected for these tiles to improve their solubility. And over here, what you can see, interesting feature, that in the case of the gold nanoparticles, which are functionalized with the tile terminated with the carboxylic group, these nanoparticles are stable in the pH range between 6.6 up to 11.3. In the case of the ammonium group, you can see that the region of the stability is between 5.3 up to 8.7. Surprisingly, if you perform the preparation of the gold nanoparticles, which will contain both these tiles with the ammonium and the carboxylic group in the ratio 2 to 1, then you can see that we can also improve the range of the pH stability. However, from the biological point of view, definitely we will be interested mainly in the stability of these nanoparticles around pH 7 or something around that value. Also, ProChimia already has huge experience in the synthesis of the variety of the tiles terminated with the several functional groups. So as you can see, we can use tiles terminated with quaternionic groups with the positive and negative charge in the same time. And also with the carboxylic group, these ethylene glycol units, which are included over here, the function of this part of the molecule is just to provide the resistance of the nanoparticle for non-specific interaction with the proteins which are available in each cell. And also appropriate functional groups present at the end can first of all improve the solubility of the final construct in the water, and also they can provide the positive or negative charge after the propanation of the carboxylic group. But also if the ethylene glycol unit will be added, in that case we can produce neutral nanoparticles. So all these properties, as you can see, combined together, they allow you for several variety of the combination of the background tile, which will provide you appropriate gold nanoparticles functionalized with the active pharmaceutical ingredient. And these background tiles can be responsible, for instance, for better internalization of these nanoparticles. All right. I would like to present two nice examples which are obtained from the Evo Nano project. We designed already gold nanoparticles functionalized with the quinolinol and with doxorubicin as well. And as you can see, TM images showed that we have monodispersed nanoparticles. DLS measurement is the slightly different method which allows you to determine the size of the nanoparticles, not only metallic core, but also the size, hydrodynamic diameter of the nanoparticles. It means the gold core plus the ligands which you have attached to at the surface. So as you can see, these nanoparticles, metallic core was around 5 nanometers, but overall, as you can see, the nanoparticles obtained after functionalization are twice larger. So it means that these tiles which we attach at the surface, they improve the size of the nanoparticles. UVV spectrum is typical like for the gold nanoparticles, so which reflects the interaction of the light, UV light, with the electrons from the metallic core. In the doxorubicin case, as we can see, the fluorescent properties for these nanoparticles can be observed as well due to the fluorescent properties of the doxorubicin itself. These two gold nanoparticles, the main advantage of the design of these nanoparticles is that, in fact, both these active pharmaceutical ingredients can be released from the gold nanoparticles in the native form. The problem with the design of the biologically active nanoparticles is that if you will use any functional group from the active pharmaceutical ingredient to attach to the gold nanoparticles, then it may happen that you will disturb the activity of the pharmacophore in the API. In that case, it may happen that the final compound or nanoparticles will be not active at all. So in that case, particularly for doxorubicin, you can see that we examine the release of the doxorubicin under aqueous conditions at temperature 37 Celsius degree and pH 2. And what you can see that the doxorubicin is released pretty smoothly. The amounts of the release of the doxorubicin you can control, first of all, by changes of the concentration of the gold nanoparticles functionalized with doxorubicin. So in fact, in this way, such construct allows to provide well, pretty smooth releasing of the doxorubicin in the long period of time. So in that case, you don't, in the case of the patient, you don't have to provide, for instance, a pill every hour, but for instance, you can provide a large dose of the nanoparticles, and the appropriate doxorubicin will be released in much longer period of time. And finally, I'd like to thank all the members of the team who contributed to the final results. So it was the work of the group when we were able to prepare these nanoparticles and accomplish the appropriate functionalization. And definitely also, I have to admit, acknowledge the financial contribution to the Evo Nano project from European Commission. Thank you for your attention. If you have any questions, I can later answer for each of them. And now, Igor, the floor is yours.
And thanks, thanks Dariusz, and thanks everyone for great presentations. I will now just briefly wrap up things and offer a glance on our future perspectives. Just a second. Okay. So after you heard all presentations and all aspects of the project, I would like to summarize and emphasize where we are now. As a result of all of these activities, we developed basically two main innovations. One is the simulation platform that at current stage can tackle numerous research issues. It can perform assessment of efficacy of different nanoformulations. It can predict effect of those treatments on tumors and on various subpopulation of tumor cells. And as you also heard about machine learning and AI algorithms, part of the pipeline is also AI-based optimization of nanoparticle formulations, and also prediction of molecular of important metrics at molecular scale, particularly surface accessible area. So that's one part. Second part, or second innovation, after all these activities, as you heard almost an hour from now, about the entire pipeline and the synthesis and microfluidics and in vivo tests from Ibane and Isabel and from Dariusz. Over those years, we streamlined the entire process of designing, synthesis, and testing and evaluating the ability of those drug carriers to be efficacious. So, as another innovation, we have something we call nanoparticle assessment platform. I will just briefly say a few more words about the tumor simulator because I missed it at the beginning, and now it is important for future development. On the right side, you have three-dimensional snapshots of tumor, because basically the entire simulation can be run either in two dimensions, two spatial dimensions, or three spatial dimensions, and each cell is a separate agent. And as I said, on the right side, you can see how tumor grows at different time points. On the right side, you can see two-dimensional snapshot after 15 days of growth of tumor with different cell subpopulations, which is quite important to what Ibane talked about, and also Sabine and Antti, about heterogeneity of tumor and our ability to represent that kind of heterogeneity in a model. So here you can see cancer stem cells, which are blue-colored, differentiated cancer cells are gray. Then snapshots of growing vascular network that in this model growth along the tumor and provide oxygen and nutrients to it. Yellow are cancer-associated fibroblasts, and orange cells that are located near the end, near the center of a growing tumor are necrotic cells. This white area in the center are already dead cells, so they are not displayed here. So that's where we are now. We can represent a heterogeneous tumor, not only composed of a bunch of same tumor cells, but we can go into quite realistic details. However, what would be the next step? As I hinted and Sabine further explained, one of the probably most useful lines of further research will be to go into development of digital twins. Digital twin is a virtual replica of a certain tumor that is reconstructed from patient-specific data. In order to do that, we would first need, in some of the next steps or next projects, to develop a data pipeline. Since we will deal with patient-derived data, that pipeline should be in accordance to all existing security protocols, so that there is no any data breach. Then, using those data, we should be able to reconstruct patient-specific digital twins. And of course, in order to make a step closer to real clinical practice, because as I said previously, we are now at the level of being able to answer to a lot of research questions, but in clinics, one of the most important issues is safety of patients. And if you want to give some recommendations, you need to be extremely sure that your recommendations will not harm, potentially harm a patient. So one of the most important next steps would be to validate those results we have using rock-hard clinical data, preferably from longitudinal clinical trials, because there we will have pre and post-treatment data, and in accordance to them we can fine-tune our model. And of course, since this field is constantly growing, constantly changing, although the current platform is already modular, we think that we should increase that kind of modularity even more in order to enable seamless incorporation of new simulations tools if it deems necessary. So in our future work in exploitation activities, we think that our value proposition could be formulated as we are optimizing the design of treatments using the digital twin of patient's tumor, and potential users of this are numerous. They can be drug and treatment companies that are developing new drugs and trying to put them on the market, so we can help in speeding up that process. Then, of course, researchers and clinical doctors who organize clinical trials, and finally, healthcare providers. If we are going to the route of digital twins, then healthcare providers who are dealing with patients would be a next obvious step. So again, thanks everyone for being with us for the next hour and a half, I think. I would now like to just make a short break, 5-minute break, and then we can go back and answer to all of your questions.
Back, everyone, and thank you very much for all those great presentations. So the purpose of these last 15 minutes is really to take your questions and also to make connections. We're reaching the end of the Evo Nano project, and it's a great chance now for us to understand what you would find useful, how we can translate some of the results, if there's connections we can make for future opportunities. So please feel free to chime in. And I've added a link to a Padlet in the chat bar, which has a number of questions that I thought we could go over, or that we'd love to have your opinion on, around the questions of what potential do you think digital twins have for nanomedicine? What challenges do you face as a researcher, as an industry, as a patient, perhaps, that you think we can help you with? What do you see as the potential for the Evo Nano tool suite? And are there bits of it that would be useful for you, and what we can do to make it easier for you to use them, or indeed to go towards personalization? And how can we facilitate collaboration going forward along the lines of what you've seen today? We have a number of panelists who didn't present today. So if everyone can put their camera on, hopefully you're able to wave when I call upon you. So Agnieszka Lindstedt from ProChimia is also here. We've also got Matimba Swana from University of Bristol, and Sepinu Azimi and Sebastian Lafont as well. So first of all, questions. There's one viewer who's been very patient, so Alexander. I'm going to call on you. Hopefully you're still there, and I'll allow you to talk so that you can hopefully ask your question live.
Can you hear me?
Yes, yes.
Okay, good afternoon. First of all, congrats. It's been extremely satisfying and stimulating, and I will have a ton of questions, but I will try to stick to 2 or 3 if you allow. Basically, I work for the TPN, the European Technology Platform on Nanomedicine, and we promote the connection of nanomedicine with other technologies. So the connection with artificial intelligence is simply extremely satisfying to hear. So fantastic seminar. Congrats to all. Several questions for, let's say, non-expert. The first one is, well, I guess that when we try to improve the design of an object, we have some, let's say, functional requirements, and we play with the design parameters. And in your optimization work and the use of AI, have you discovered that we don't have, in fact, like the classical research lab, that the classical small company doesn't know which are the real design parameters they can play with? Is the size, the charge, the dispersion, the surfactants, etc., enough, or have you discovered something surprising, something which is missing in the scheme? That's my first question. The second question is, what would be the most counterintuitive or the most surprising results that you would mention along this use of artificial intelligence? What did artificial intelligence find that you all could not find without it? And maybe my last question would be more like, did you find also some results that could apply to out of the pure drug delivery for nanomedicine, like for instance, how to enhance the effect on radiotherapy or out of cancer? Thanks a lot.
Great, thank you. So about the functional requirements, and was there anything surprising or any parameters that we haven't thought of on the functional requirements? I should say to our panelists, raise your hand as well if you'd like to add something to these answers. So I think there's multiple aspects. One is that the parameters that we chose are just a subset of things that you can change on nanoparticle design, and there's levels of abstraction. So at the level we've been looking at, it's quite abstract in terms of the size and the shape and the charge, et cetera. And already those sort of high level parameters give you surprising results. So that example I gave where a particle has a high binding affinity, which sounds good at the individual level, because if you test it in a Petri dish, you have a really good uptake of those nanoparticles within those cancer cells, performs really poorly at the level of the tissue. And so I think thinking sort of systematically about those parameters and what they mean for the collective often throws up surprising results, which are only surprising if you haven't thought about them, but you have to do that process of simulating them to be like, oh, that makes sense. And then you think slightly a bit differently about what you're doing. At the molecular scale, Igor, I don't know if you want to comment on the tumor scale.
Yeah, I just wanted to add a molecular scale, because that's something we didn't initially thought about. And now, as Sabine said, it seems quite obvious, but before and at the level of project preparation, yeah, we didn't thought about that. Then we figure out that something is missing and something important. Yeah, I guess that would go under surprising finding. To what extent is molecular analysis of surface accessible area important in determining potential functionality of those nanoparticles? That's what Oksana talked about.
Yeah, about how was the AI useful? I think it's quite interesting, or what came out of that you wouldn't have been able to sort of wrap your head around otherwise. In the cancer stem cell strategy that we had put, the Evo Nano platform paper has sort of toy scenarios that we can reason about, so that we can somewhat validate whether we think the results make sense. So, for example, with the cancer cell strategy only, that particle that emerged from artificial intelligence is actually very similar to what's been designed with Doxil, which is an approved nanoparticle. So that was helpful for that paper as a sanity check. And I guess it was good news that the particles that they're designing have some of the features that we also observed were useful, that came up through the evolutionary process. For the cancer stem cell one, there was a particle that we expected, and then there was a particle strategy that we didn't expect with a bigger nanoparticle. And that one was counterintuitive. And it's typically the case where it theoretically would work, but that's not clear whether it would work in reality. And we would need to validate that with experimentalists. So it gives you good hypotheses, things that we haven't thought of, things that don't currently exist in the clinic, that we can give back to someone and say, would this particle actually work in that context? Antti, anything surprising on your strategies?
Yes, although I briefly introduced in my presentation. So although we're using a high level with high abstraction, as Sabine said, simulator, the PhysiCell, and we did some changes without actually testing them in clinic or in vitro yet or in vivo. So we found that using multiple nanoparticles without extensively simulating their collective results. So when we apply them at different phases of the growth of the tumor, we can have better results, because different types of nanoparticles may target different areas of the tumor, and even the tumor didn't have the whole range of different cells that Igor showed in the last presentation. Just having a homogeneous tumor can be with one type of cells, but because some of them collapse, some of them die because of one drug, they form different groups that behave a bit different and go away from the nanoparticles or something like that. So that was not that much surprising, but not expected with the level of abstraction we are bounded to.
And that's a good point, as well. As you complexify these scenarios and allow for different types of particles interacting, you have the potential for new strategies that are quite different, actually, from what people currently consider.
May I admit something? Well, from my point of view, I was surprised that not only the API is important in the case of the gold nanoparticles, but also a tile background which we have used for the construction of the gold nanoparticles. Ibane can confirm that in the case of the background tile, in some cases from API, the internalization was very poor. When we switched to another background tile, the internalization was pretty good. And the problem only is that if you change the API, the same background is not able to work efficiently. So it looks rather that each API requires individual development of the background tile, which will provide good internalization. So there is no simple answer for the final properties. It's really complex. So without computation methods, I think it's like a black box. We cannot find quickly and efficiently appropriate background tile. But if we combine the comparison of the biological studies with computation, I think in that case, the synthesis can be performed in the rational way, and the design of the structure of the nanoparticles can be much more efficient in this way.
And the third question was about enhancement of treatments like radiotherapy and chemotherapy. Was that one about combination therapies?
I was more thinking about, you know, that there are nanoparticles out of the drug delivery, let's say, gold way against cancer. Some of them are used to enhance the effect of X-rays, the delivery locally in the tumor, or even nanomaterials are used for other things. So have you discovered some things that could be useful out of drug delivery?
Oh, that's interesting. I don't think we've looked at that, but fundamentally we have a way of predicting where the particles go. So if for imaging it's important to accumulate in certain areas of interest, then that's something that you could in theory use the models for.
That would be of great interest.
In the case of irradiation, frequently you need some kind of the synthesis, compound which will make this much more sensitive interaction with the irradiation. So
yeah, like hafnium.
Yeah, so we didn't use such compounds, so we do not have any experience in that area. So, but probably it's possible, but the only problem is that the final construct may be simply too large for internalization. And also we have to consider that the internalization should be selective. So healthy cell shouldn't internalize these compounds, but only cancer cells are supposed to internalize. So the selectivity of the internalization is another problem which should be solved in the future.
Great. Well, thanks very much for those questions. Did you have any other ones? And it's good to have you here.
Can I?
If anyone else has a question, please go ahead and put it in the chat. But otherwise, I think you're free to go. Yeah, another question, sure.
It will be my last one, I swear.
That's good.
Yeah, well, let's call it enthusiasm. Okay, so we had a very complex challenge, and you try to bring some kind of smarter design, but the smarter design can come with even more complex characterization. So have you found some kind of critical pathway for characterization? And very importantly, because I truly believe that we need to put more synergy between European projects, have you already discussed about your results with the REFINE project? And if not, would you be, let's say, keen on trying to discuss with them? We can create this connection very easily at the ETPN.
Ibane, do you want to comment on characterization, and maybe then we can comment on how it's tricky to match up as well with computation?
So do you want me to answer this?
Yeah, sure. I think on the characterization side.
So yeah. So as Dariusz said previously, it's difficult to put a general rule for all type of nanoparticles. For instance, in the case of gold nanoparticles that we were working with, so the zeta potential, the surface charge, and the size are the most critical parameters. And with these two, we can do many things. But for other nanoparticles, those are not that important. So it's difficult to establish a general rule. It depends a lot in the type of nanoparticle. And then I completely agree with you that it will be good to have this interaction with the REFINE project. I'm not involved in that project, but I know other collaborators that are there. So it would be good if we could, get in contact. Yes.
Yeah, I'll try to help, because they've just released some kind of decision support system with some kind of scoring. You can decide many parameters about your nanomedicine product and select a critical pathway for characterization. And I guess that you would be of mutual help, and you are close to the end of your project, so you are very mature. So maybe the ETPN can try to be the middleman here. So let's keep in touch on that, if you agree.
Sure.
Great. And on the characterization side, it's also very different what we need in simulation to what's usually done in the lab. So that's also something that we're learning is we have ideas of where the particles go, but we would love to zoom into the real tumors and see where the nanoparticles have accumulated. And that's always tricky to quantify.
Thanks a lot for all the answers. Thanks again.
Thank you. Yeah, thanks very much for joining. We have a question from Sangeeta. You should be able to speak now.
Hello, ma'am. Hello.
Yeah, we can hear you.
Am I audible, ma'am?
Yeah, very good.
Yeah, I'm a scholar from India. I'm very happy to participate in such meet. And I'm working on cancer, lung cancer with nanomaterials, medicine, whatever you call. And I need to take it like in future, I want to convert it like a kit. So I would like to get some advices or any ideas, parameters that I should follow in future to make it as a kit to detect the biomarker. I mean, like early detection of lung cancer biomarkers. That's my topic too. So I would like to get some suggestions from the expertises. So
okay.
Please.
And when you say a kit, are you looking for an actual nanoparticle at the end?
Yeah.
Or, okay, right. And for a biomarker, you want them to accumulate in the lung, or you want them to be able to detect a biomarker from the lung?
I would like to say something like pregnancy kit, for example.
Ah, I see. Okay,
Yeah.
okay. So a
like
test.
I would like to develop, for the early
Right.
detection. I'd like to develop a kit like that. So I need some suggestions and advices from the scientists in our meet. No.
Yeah, so
I'm happy to
really,
get such points.
That's really interesting because we have a project with Cancer Research UK about designing biomarkers for blood for brain tumors, and the models that we've built. Imagine this, now you inject a nanoparticle, it goes through the bloodstream, ends up in the tumor, but a biomarker does the opposite. It starts from the tumor, exits the tumor, ends up in the bloodstream or whatever you're trying to collect the sample with,
obviously.
And then you've got that in there. So actually some of the models we're doing, if you do the reverse, you get a biomarker model that you might be able to use for that. On the nanoparticle design, I don't know, ProChimia, if you've got any insight on how to design probes for biomarkers?
No, we do not have experience with biomarkers.
Okay, okay. You could think about decorating them with the right ligand to target some of the biomarkers.
There are so many interesting features to take care of that we do not have.
Fair enough. Does anyone else have advice?
Thank you so much.
Okay, great. So if you haven't contributed to the Padlet and you're unfamiliar with it, feel free to have a go in your own time. You just add a little plus sign and you can add some thoughts, or you can email us. All the contact information should be on the website. So Igor, do you want to close? I think we're probably at the time.
Yeah, thank you.
Thanks, Sangeeta.
Thank you.
Yeah, thanks, Sabine. It was quite interesting. We made an overview of what we did, and yeah, for me, the last part about lessons learned and what was surprising was quite interesting, because during the project I was always struggling with that question: why is this happening? Why is that not happening? And so on. So it is quite an interesting set of results, and it opened more questions than probably answered, which is, I guess, great, because it opens new avenues for future research. So yeah, I think we are on a good track to try to move it further towards digital twins and improve even better characterization of nanoparticles, because that's quite important here. So yeah, thanks.
Okay, bye everyone.